So hi, hello and welcome again, Microbehunter here, and today in this video I would like to explain to you Brownian motion because I did receive an email, so it's a little bit of a special requests video that I'm making right now. I would like to read out the email first, and then I'm going to respond to it by essentially also explaining a little bit of what these little white dots are here in the background. I hope you're doing well. I love your videos. You not only share beautiful footage, but also explain things in a simple, easy to understand language. Thank you for making this accessible to everyone. I would like to, of course, return the thank you to you, and I'm happy that the videos are appreciated. I'm an undergraduate student, and we recently did a physics lab experiment on Brownian motion using silica microbeads. Then we tilted the microscope to change the Brownian motion into more direct motion or directional motion. Could you make a video showing this kind of directed particle movement by tilting the microscope? I'd love to show it to my class if you do. And then kind regards. Well, thank you, of course, for the question. And sure enough, here we are. And what you see over here is basically those white dots that you see here. These are individual cells, algae, and from a water sample. And if you look very carefully, then you're going to see that some of those algae, let me focus here, they're vibrating a little bit. And this vibration, that is called Brownian motion. I'm going to switch on the arrow right now. It's because I want to show you a couple of other things here as well. So if you look very carefully, then those tiny dots here, those cells, they vibrate randomly. And the reason is because they are surrounded by water, and the water has a temperature, and the water molecules are vibrating, and then they bump into those cells, and this causes the cells also to move around in a random manner. And that's called Brownian motion. However, that's not the only thing that you see here. If you look, and let me refocus, there are some of the cells seem to behave a little bit differently, especially if you look over here. They seem to, be a little bit moving a little bit on their own, right? Oh, over here, this one here in the corner. You see this one here? I'm going to recenter this. Here we go. That's a gigantic air bubble that you see up here, right? So that's clearly directional motion, but many are because they have a flagellum that, or cilia that move, and it kind of moves them forward. But you see that many of the other cells are moving and vibrating randomly, and this is essentially Brownian motion, and it's non-directional. When the cells move on their own, then this is of course a directional movement. However, the request was, can you tilt the microscope? I cannot do that, but I'm going to show you a different way. The reason why I cannot tilt my microscope to make the liquid flow into one direction is because my microscope is simply too heavy and I don't want to damage it. However, there is a little workaround, and actually there's several workarounds here. And one way would be is that you simply take a pipette and you add a little bit more liquid, or you take some tissue paper and you remove some of the liquid, and this causes a certain directional movement and a streaming motion, and then you have the random Brownian motion superimposed on that. So I want to show this to you. So I'm just going to grab a little bit of liquid here from my jar, and I'm going to add it now to the corner of the cover glass, and let's see when it is you see soaked in by capillary action. Right? Then the liquid spreads beneath the cover glass in the microscope slide, and you see, of course, that those particles, those cells, also move, depending a little bit on the capillary movement of the water. Maybe there's some air bubbles that are kind of collapsing. So you see it's a little bit, directional, but you see that the vibration of the Brownian motion is superimposed. So what I'm going to do is I'm just going to go down with the magnification a little bit to give you a better overview. This here is now, you see the dots are now very small. And what I'm going to do now is I'm going to take a little bit of tissue paper, and I'm going to remove some of the liquid again. And this should also induce again a directional movement. So I'm just going to touch with some tissue paper the edge of the cover glass. Of course, I'm kind of, you see the air bubble also increasing in size, pushing the particles away. And you see then there's also a streaming motion going on. But at this low magnification, of course, we do not see the movement of the individual cells, the vibration, the Brownian motion quite well. So we have to go up a little bit. And if you kind of wonder why the color here is different, that is because I'm using a special technique called DIC, differential interference contrast, and this gives it a little bit some of the colors here. But I think we're going to go up again with the magnification to have a look a little bit in more detail. Yeah, this is now at a high magnification. I'm using now a 60 times objective, and of course, the higher that you magnify, the more visible the Brownian motion, right? So, however, those cells are fairly large, right? And this means they have a lot of inertia, and this means they cannot move quite as easily. And the larger the object, the more inertia it has, and the more stable it is. And for this reason, I think we have to look at some particles that are yet significantly smaller. And what I'm going to show you now is highly diluted milk. Just regular milk. So a few drops of milk in water, because milk contains a lot of fat droplets, and those fat droplets are very, very small, and therefore they also move much more. And I would like to show that to you. Why diluted? Because if you just take concentrated milk, then there's so many fat droplets, that essentially the movement is inhibited. But if you dilute it down, it's going to be much more visible. I'm going to show you. Look, here's a ciliate. Yeah, that's also an example of directional movement. Okay, and then at the end of the video, I'm going to talk a little bit about the random movement, and also called kinesis and taxis in microorganisms, because it also has to do something with movement, but it's again a little bit different. But actually, it also kind of fits into this topic here. Look, another ciliate over here. Okay, so now let's have a look at some milk. So this here is now some milk, and this huge thing here, that is just a contaminant, some kind of a fiber, which happens to be on the on the microscope slide, because I wiped the microscope slide, so maybe it's a little bit of some cloth or some fiber from a tissue paper. But those little dots that you see here in the background, right? These are fat droplets, right? And they also vibrate, and they also seem to be streaming a little bit, because as the liquid evaporates from beneath the cover glass, this also causes this streaming motion. And look. Again, a directional movement, usually when some kind of a bubble collapses somewhere, or two bubbles combine, and this can cause a directional movement and a streaming motion here, right? So, but let's move the arrow a little bit out of the way again. But because those fat droplets are significantly smaller, they also move, not move, but their Brownian motion is a little bit more pronounced here, right? So, if you just look around here, again, look, an air bubble and some kind of a contaminating fiber, it doesn't really matter. Sometimes if you've got two air bubbles next to each other, you can actually see how those fat droplets kind of stream around the air bubble as well. So one of the things that I could try is I'm just going to take my tweezers, and I'm going to now press down on the cover glass itself, and maybe this will also induce some kind of a directional motion. So I'm going to quickly press down on the cover glass. As you press down, then you're pushing the liquid away, and then you can of course also see that this is directional. So I think you get the point. What I would like to do now is I would like to go back here, and maybe I'm going to find again a ciliate. I would like to talk a little bit about the random movement of microorganisms and the directional movement of microorganisms. So basically when they move on their own. It has nothing to do with Brownian motion as such, but it's also interesting because you also find both of these cases in microorganisms. Okay, so look, that's a tiny one. It's a flagellate. I'm just going to point the arrow. This tiny guy over here, right? Maybe you see this tiny extension that it has. This is called taxis. Microorganisms move on their own, also directionally towards a favorable condition. More oxygen, for example, towards food, away or towards light, and this is kind of a directional movement, and it's called taxis. However, in this case, taxis is only possible if you have the stimulus approaching from one direction, right? So, for example, if there's light shining from one side, and the organism, let's say certain algae, then they want to move towards the light, right? But what happens now if there is no directional stimulus? Let's say, for example, a whole area is illuminated. Let's say the light is shining from the top, and this is not a favorable place. How would the microorganism then know into which direction to move? Because the stimulus is not coming from somewhere. And here you have again random motion. It's called kinesis, and what they do is the following. And I'm just going to trace this out now with my arrow that you have here. The microorganism is simply going to move around on its own randomly. So again, this is not Brownian motion, right? It's much more than that, because they're using their flagella or their cilia to move around. And if the condition is not favorable, then what they will do is they will run a longer distance and then basically turn randomly and then again run a longer distance. So if it's not favorable, they will start moving randomly around like this, right? And the more favorable the conditions become, the shorter the runs and the more frequent the turns are, right? And mathematically, when this happens, is then they will move actually statistically towards the area that's more favorable. So let's say there is some kind of a gradient of something, let's say an oxygen gradient, right? And there's more oxygen here in the center than on the side, but the stimulus is not coming from a particular direction. It's just generally like that. So what they will actually do is as soon as they reach a more favorable condition, then they will simply start moving around with shorter runs and more frequent turns, and then they will tend to stay in that area longer, right? So what you have is, and why I like this so much, this system, is because it kind of shows that randomness in movement of microorganisms kind of also can result in some kind of a directional movement, so to say, that they statistically move towards an area which is more favorable, right? Simply by reducing the more favorable it becomes by reducing the runs and by increasing the turns, right? So this is simply an example also from biology, Brownian motion being this random movement from physics primarily, and then you see also random movement and directional movement in biology, and that's essentially the connection that I wanted to make here. And here you can see again over here, this is clearly a directional movement, because I suppose because the water is evaporating from the edge of the cover glass, and therefore there it's kind of streaming into one direction. Or it could be that there is simply more, now it stopped, and this happens quite frequently somewhere where an air bubble kind of pops or fuses together, or can be several possibilities. By the way, in case you're wondering, changes can also happen because when algae produce oxygen by photosynthesis, that's clearly directional movement here. When algae produce, then bubbles can appear, and they can also push the particles around, right? So that's a ciliate, right? Very clearly directional movement, I think. However, maybe also in combination with random movement, because it does turn around, and if it is able to find a more favorable condition, right, then it will turn more frequently. So you see, you have both a directional and random movement kind of combined. So as a matter of fact, those ciliates are able to also sense differences in concentration. So, for example, when one part of the cell is exposed to higher oxygen concentration than the other part, then they're somehow able to sense gradients, chemical gradients as well. Okay, I think that's going to be enough. I've almost talked 14 minutes on this. I hope it was informative. I want to thank you again for the question. I'm always happy for suggestions to make videos. I wish you all the best and happy microbe hunting as always. And hope to see you again in the next video. Bye-bye.